Good morning and happy Friday to all. My name is Camilo Gonzalez Velasquez, and it's a pleasure for me to co-host today's Thyroid by MD's webinar. We're truly honored to have two young and exceptionally talented thyroid specialists with us today. Our presenter, Dr. Iñigo Landa, a renowned figure in thyroid cancer genetics and biology. Dr. Landa serves as an associate scientist in the Division of Endocrinology at Brigham and Women's Hospital and is an assistant professor of medicine at the Harvard Medical School in Boston. His career in cancer genetics includes postdoctoral training in Dr. James Fagan Lab at Memorial Sloan Kettering Cancer Center and graduate studies in molecular biology in Dr. Mercedes Robledo Group in the Spanish National Cancer Research Center in Madrid, Spain. Dr. Landa's work is key in deciphering the mechanisms behind genetic events in thyroid cancer progression. Today, he will share his recent publication in JAMA at Biological and Clinical Implications of Genomic Alterations in Thyroid Cancer. We're also joined by Dr. Pinar J. Smith, a dear friend and distinguished colleague who will help me drive discussion at the clinical aspects of Dr. Landa's lecture. As a director of the Thyroid Cancer at Rush University Medical Center in Chicago, Dr. Smith's focus in thyroid and parathyroid disease has led her to spearhead tumor boards that enhance multidisciplinary patient care. Currently, she contributes her expertise as a member of the American Thyroid Association Surgical Affairs Committee and the editorial board of the Clinical Thyroidology for the Public Journal. Her path in endocrinology was profoundly shaped by a fellowship in the Icahn School of Medicine at Mount Sinai, Beth Israel, under Dr. Terry Davies. Additionally, she was on the staff of Columbia University Thyroid Center in New York for 3 years prior to joining Rush University Medical Center. We're immensely fortunate to welcome both Dr. Landa and Dr. Smith today for the webinar. Their combined expertise promises an incredibly interesting session. Before we begin, I would like to invite all of our audience to send in questions that we will try to get to as many of them by the end of the hour. So with no further ado, Iñigo, please. The mic's all yours.
Thank you. Hello, everyone, and thank you for the kind invitation. And thank you, Camilo, for the nice introduction. All right, so I have nothing to disclose. So this is the presentation outline for today. I would first be speaking about the genomic and biological landscapes of thyroid tumors, and then I will be showing some examples on how genomics can inform the management of thyroid cancer patients. I will be primarily quoting work from others, which we compiled in this recent review published in Nature Reviews of Endocrinology. I will primarily be speaking of 3 concepts, which I think are interrelated. First, the identification of genetic alterations based on the breadth of genomic sequencing of patients' tumors. Second, the idea of understanding the mechanisms behind those mutations that drive thyroid cancer initiation and progression. And finally, the application to clinical practice. And these 3 concepts are typically linked in this direction, from genetic alterations to mechanisms to applications in clinical practice. But I think there are connections in every single direction, and I'll try to make those connections today. This is, I think, this is the picture of cancer genomics in general and thyroid cancer genomics in particular in the last decade or 15 years. I think we are today in 2024 in a situation in which in the last decade or so, with the implementation of sequencing technologies, we were basically buried in a ton of genomic data on genes that have been associated to thyroid cancer. And obviously, by definition, understanding the mechanisms and applying that knowledge to the clinic lag behind, but we are beginning to close this gap here. So I will be primarily speaking about follicular cell-derived thyroid cancers, in particular differentiated thyroid cancers, papillary or PTC, the most common thyroid cancer subtype, as well as follicular. And I will also mention advanced or aggressive thyroid cancers, namely poorly differentiated or PDTC, and anaplastic thyroid cancers. The most important part of this slide is this arrow here, because I think it informs most of what we do in the lab and what we are trying to apply in the clinic. And it's the idea of most advanced thyroid cancers likely evolving from pre-existing well-differentiated counterparts. Now, a little bit of broad view of cancer genomics. This is a comparison of the mutation burden across multiple cancer types. And I have highlighted here skin melanoma in yellow, which is one of the most hypermutated thyroid cancers. And in blue here, we have the papillary thyroid carcinoma. And you can see that comparatively, PTC has a very low mutation burden, and this helps quite a lot at establishing genotype-phenotype correlations. I highlighted melanoma not only because it's the most hypermutated tumor type, but also because the mutations that occur in melanomas are quite similar to those in thyroid cancers. Now, what are the genomic determinants of thyroid cancer initiation? So the first thing that you need to keep in mind is that thyroid cancers, or let's say 80–90% of thyroid cancers, are initiated by overactivating, by constitutively activating this pathway, the MAP kinase signaling pathway, which is a key process for cell proliferation. And thyroid cancers do so by mutating usually one of these genes, either BRAF, typically a V600E, or RAS genes, NRAS, HRAS, or KRAS, or via gene rearrangements, activating one of these receptor tyrosine kinases, typically RET, and in other cases, NTRK1 and 3, ALK, and others. All of these alterations are mutually exclusive, meaning that just a single alteration is sufficient to activate MAP kinase pathway and to initiate a thyroid cancer, usually a PTC or an FTC. Now, we know this from multiple sequencing studies. I think the flagship study is the one by the TCGA, the Thyroid Cancer Genome Atlas, in which they applied multiple omics platforms to a series of around 500 papillary thyroid carcinomas and defined their mutational and transcriptomic landscapes. In this figure here, you can see the 500 thyroid cancer patient tumors here, and then the top mutations in different genes in the vertical axis. So one of the main conclusions from this study is the mutual exclusivity nature of BRAF, RAS, and RET alterations, and you can see this ladder pattern here. Around 60% of PTCs have a mutation in BRAF V600E, 13% of those in a perfectly non-overlapping fashion mutate RAS genes, and then down below here, we have the fusion oncoproteins, including RET, NTRK, and a few others. The TCGA also defined expression signatures, and I will briefly comment on two important ones. The first one is what we call the BRS, or the BRAF V600E RAS score, which is a proxy of MAP kinase output. And it's relevant not only because it classifies BRAF V600E and RAS mutant as BRAF-like and RAS-like, but also because it allows us to classify thyroid tumors driven by other mutations into these two transcriptomic categories. For instance, we know that mutations in BRAF other than V600E, for instance, K601, are RAS-like, and that has some signaling consequences and phenotypic consequences as well. The TCGA also defined this 16-gene transcriptomic signature that they call the Thyroid Differentiation Score, or TDS, a measure on how well-differentiated these already well-differentiated tumors are. And it was this correlated inversely with the BRAF-RAS score. So BRAF-like tended to be less differentiated than RAS-like. So we are beginning to call thyroid tumors BRAF-like and RAS-like more than BRAF mutant or BRAF V600E mutant and RAS mutant. And this, in turn, correlates pretty well. These are not absolute, but they are quite good with the histological subtypes. We now know that BRAF-like thyroid tumors, differentiated thyroid tumors, are typically going to be classic or tall cell variants of PTCs, whereas RAS-like tumors tend to produce follicular thyroid cancers or the follicular subtype of papillary thyroid cancers. It's a nice example in which the driver connects two entities that used to be farther apart in previous classifications, and they are becoming more and more or closer and closer. Now, there's an important signaling consequence in BRAF or BRAF-like versus RAS-like tumors, and is the degree of activation of the MAP kinase pathway. So BRAF V600E activates the MAP kinase pathway to a higher extent than RAS-like or RAS mutant tumors. And this results in a higher ERK-mediated transcriptional program and in a lower differentiation or expression of the genes typically involved in thyroid differentiation, including NIS and important genes in thyroid metabolism and iodine processing. So this is going to have some important clinical consequences that I will comment later on my talk. The potency of the different potency of BRAF and RAS in thyroid cancers was actually proven early on in the first or one of the first waves of mouse models of thyroid cancers, 15, almost 20 years ago in some cases, in which engineering the expression, the thyroid-specific expression of BRAF V600E almost invariably showed or resulted in papillary thyroid tumors with a high penetrance. RAS oncogenes, NRAS, HRAS, KRAS in mouse models, genetically engineered mouse models, also showed a weaker transformation potency, typically given follicular adenomas or even follicular thyroid carcinomas, follicular variant of papillary thyroid carcinomas with a long latency, or just thyroid hyperplasia after, a long latency as well. So that was, pointed from the very beginning with basic science approaches. Now, there are a few exceptions. I'm going to mostly be talking about adult sporadic papillary thyroid cancers, but I want to acknowledge the diversity of other thyroid tumors. For instance, pediatric thyroid cancers are clearly enriched in fusion oncogenes. Something in between 35 to 60% of pediatric thyroid cancers are initiated by a receptor tyrosine kinase fusion, RET, NTRK3, MET, and others, versus around 10% of adult tumors choosing to mutate these kind of genes. Another specific type of thyroid cancers in which fusion oncogenes are very prevalent are those linked to radiation. And the best example here are those thyroid cancers that arise around the population in the Chernobyl area after the nuclear plant accident. And in this paper published in Science a couple of years ago, they nicely established, they estimated the amount of radiation that patients in that area received and how it correlated with the frequency of fusion oncogenes in their thyroid or driving their thyroid cancers. Aside number 3, follicular thyroid cancers, I briefly mentioned them already, but you can see that pretty much BRAF V600E mutations are absent in FTCs, and instead RAS mutations and a few other oncogenes are driving these tumors, and in terms of expression signatures, they are typically RAS-like, and that makes them closer to the follicular variant or the follicular subtype of. Last exception, oncocytic cancers, formerly known as Hürthle. I think this is a nice example on how an unusual phenotype also correlates with unusual genotypic features. As you know, oncocytic cancers have this very eosinophilic look under the microscope, and this is the result of the accumulation of mitochondria in the cytoplasm of these cancer cells. And a couple of back-to-back studies in 2018, one from Dana-Farber, one from MSK, prove that these cancers are very unique. They mutate genes from the mitochondrial genome, in particular respiratory complex one, and they accumulate homoplastic dysfunctional mitochondria. And in terms of the nuclear genome, there are some genes mutated there that also occur in other cancers, but the main characteristic is a near total haploidization of the genome. Pretty much with the exception of a few chromosomes, they maintain only one copy of the rest of their genomes. And we're beginning to understand what this means for clinical practice and other topics, but these tumors are very unique. So first recap from the genomics of thyroid cancer initiation or of differentiated thyroid cancers. I think it's fair to say that most thyroid cancers are initiated by a single genetic alteration, which activates the MAP kinase signaling pathway. BRAF mutant or BRAF-like and RAS-like activate MAP kinase pathway to a different degree, and this determines their predominant histologies, the level of thyroid differentiation, and a few other features of these tumors. And finally, fusions involving receptor tyrosine kinases, mostly RET and a few others, are alternative driver events. They tend to be BRAF-like, and they are particularly enriched in pediatric and in radiation-induced thyroid cancers. Oncocytic tumors are phenotypically and genomically distinct thyroid tumors. Now, I will now briefly summarize the genomic determinants of thyroid cancer progression. Again, I took this figure from a preprint that was posted a few months ago. It's a paper that I'm assuming will be published soon, in which they characterize the genome of anaplastic thyroid cancers. And here I'm highlighting again skin melanoma and papillary thyroid cancers. And if you look at ATCs, remember one of the most aggressive tumors overall, well, they are hypermutated compared to PTCs. But if you look at the global picture, they have a relatively low mutation burden. Again, we are able to establish connections even in these very aggressive tumors. So, as I mentioned, the main signaling hub for thyroid cancer initiation is this MAP kinase pathway via mutations in BRAF, RAS, or RTKs. For progression, cancers diversify the pathways they significantly altered. Not all of those at the same time, usually one or two on top of a MAP kinase pathway alteration. In other words, thyroid cancer initiation is very linear, MAP kinase driven in most of the cases. Thyroid cancer progression is much more diverse in their genetic mutations and in their cancer biology. And some of these other pathways feed into the MAP kinase pathway as well. So we know this also based on multiple sequencing studies on advanced and poorly differentiated and anaplastic thyroid tumors. I will briefly show you what we did when I was back in MSK. And we characterized the cancer genome of 84 poorly differentiated and 33 anaplastic thyroid cancers. And again, this is the oncoprint of these tumors. The 84 PDTCs are on the left block, and the 33 anaplastics are on the right. And here I'm listing the main genetic events. Few take-home messages from this study. First, that the mutation burden correlates with aggressiveness. Anaplastics are hypermutated compared to poorly diffs. Second important point is that most of these tumors maintain a truncal mutation in either BRAF, RAS, or RET in a few others RTKs, strongly pointing at an evolution from differentiated to undifferentiated thyroid cancers. And they obviously, as I mentioned in the previous slide, mutate other genes. PIK3CA, PTEN, some of these occur in a very specific fashion, as well as IFAKs, for instance, typically associated with RAS mutations. And some others are less picky in terms of tumor driver, TERT, and P53, which is particularly important in anaplastic thyroid tumors, occur across the board here. We also identified some alterations that cluster in key pathways and functional groups, and compare their prevalence with the same gene mutations in papillary thyroid cancer from the TCGA. And you can see that poorly differentiated and anaplastic in particular are enriched in mutations of effectors of the AKT pathway, of members of the SWI/SNF chromatin remodeling complex, also genes encoding histone methyltransferases, as well as genes involved in DNA repair via this mismatch excision repair pathway. So there are obviously many other studies of cancer genomic or characterization of the mutational landscape of tumors, and in particular advanced tumors. And we summarize those in this figure from this recent review. Each of these dots is a different patient cohort. And we listed the main thyroid cancer subtypes and the prevalence of specific genes here, P53, third promoter, P16, etc., and some of these pathways and functional groups. And you can see, for instance, if you focus on the red bars, which represent the anaplastic thyroid cancers, and the light blue representing the papillary thyroid cancer, that there is a clear increase in prevalence of some of these mutations as these tumors become more aggressive and less differentiated. So how to put all of this together? We came up with this idea of this marble run representation, in which each of these marbles or balls represent a tumor, and they are color-coded by their main drivers, BRAF, RAS, RET, etc. And as they progress, as they evolve, they go down this slope of de-differentiation and increased aggressiveness. And you can see that within this evolution, probably because of their genomic simplicity, each of those tumors, and we are only representing, the most frequent cases here, follow on a specific track that I number here 1 to 6. So for instance, here I'm representing a BRAF mutant, RAS mutant, and RET rearranged differentiated thyroid cancer. You can see that we are in the papillary and follicular thyroid cancer, like well-differentiated space. And within those tracks, as we commented before, BRAF associates with classic or tall cell PTCs. RAS mutant tumors are typically the follicular subtype of PTC or the follicular thyroid cancers, and so on. The additional mutations here are represented by tags, and in some cases they associate with specific drivers. For instance, the BRAF and PIK3CA mutations tend to co-occur. Same with RAS and mutations in IFAX, and those also determine specific phenotypes. RAS and IFAX typically progress to poorly differentiated thyroid cancers. BRAF and PIK3CA are usually associated with this newly defined differentiated high-grade thyroid cancer, and they can evolve all the way to anaplastic thyroid tumors. And then finally, there are other mutations. P53 is a prime example that seem to fast-track tumors of any nature towards strong de-differentiation and progression to anaplastic thyroid cancers. So, in summary, from this part, the genomics of poorly differentiated anaplastic thyroid cancers, I think data support a model of tumorigenesis in which poorly differentiated anaplastics arise from well-differentiated tumors through the accumulation of key additional genetic defects in specific pathways. PDTCs and ATCs differ in their genomic features as well, and some of these features have clear diagnostic and prognostic implications, including for the well-differentiated counterparts. So now, for the second part of my talk, I will provide a few examples on how genomics can inform the clinical management of thyroid cancer patients. I'm going to start with a couple of examples on prognosis. And for that, I'm going to briefly comment on the role of TERT as a biomarker of poor prognosis. As we discussed before, mutations in the promoter region of TERT are relatively infrequent in papillary thyroid cancers of the TCGA, much more frequent in poorly differentiated, and very prevalent in anaplastics. And in all three groups, they tend to co-occur with either a driver mutation in BRAF or in one of these RAS genes. Now, the first study, remember, these mutations were discovered in melanoma only 10 years ago, and shortly after reported in thyroid cancers by several groups. I think this is the first study in which they performed a retrospective analysis of papillary thyroid cancer patients, and they associated the third promoter mutation with an increase in metastatic and persistent disease in PTCs. Probably the strongest results in terms of indication of poor prognosis come from studies combining BRAF and TERT. I think this genetic combo is a very good biomarker of poor prognosis in papillary thyroid cancers. And here they show that in these Kaplan-Meier curves for all PTCs and for classic PTCs, and how this, as you can see, this red line is significant compared to either of those mutations alone or those PTCs that were wild type for both. And they associated this to a decreased recurrence-free survival in this patient cohort. Most of the studies on TERT have been performed on BRAF mutant thyroid cancers. There are a couple of reports also associating the prognostic or pointing at the prognostic power of TERT in RAS mutant disease, and also associating that with decreased disease-free survival in differentiated thyroid cancers. Because these were RAS mutant, most of these were follicular thyroid cancers or the follicular variant. This is a summary slide of the many studies associating TERT promoter mutations with thyroid cancer prognosis, and it's already outdated. It's a review from 2016. There are at least another 10 studies in the last 8 years or so. I'm gonna give you a flavor of what we are doing on the basic science front to tackle or to understand what TERT reactivation means for thyroid cancer progression. And this is something that we published recently, in which we use a mouse model of thyroid-specific BRAF mutant, BRAF V600E mutation. And on top of that, we generated via CRISPR a TERT promoter mutation. And we also used a previously published model of TERT overexpression. And when we looked at the tumors from these animals, all of the BRAF mutant animals generated papillary thyroid cancers, which did not progress. But around 30–35% of those animals with telomerase reactivation by either of these methods progressed to poorly differentiated thyroid tumors. A small subset of animals that we aged to up to, I think, 40 weeks started showing these ATC-like phenotypes with spindle cells, infiltration by immunosuppressive cells, etc., exclusively in those two models with telomerase reactivation. We also we did RNA sequence on these tumors from our mouse models, and we were able to identify non-canonical functions of telomerase, specific pathways, some of which point to well-known pathways here, and some others that point to cytokine and chemokine signaling, probably as a crosstalk with the immune microenvironment, being overexpressed in these thyroid cancer cells of slightly more advanced phenotypes, and in a way, giving us some hints on some of the pathways that might be important in TERT reactivated thyroid cancers. And some, this is obviously preclinical research, but eventually we could try to target some of those pathways. Now, speaking of using genomics for prognosis, I'm going to show you another recent paper from the lab. Here we studied a cohort of 225 BRAF V600E mutant papillary thyroid cancers, and using the OncoPanel platform here at the Brigham and at Dana-Farber, we classified these tumors based on the presence of other oncogenic mutations in a highly curated list of genes that we know are important in thyroid cancer biology, and in particular in thyroid cancer progression. And we divided our cohort in those who in three groups. One group in which only BRAF V600E mutations were present, and another group in which additional alterations were present. And we were particularly interested in mutations affecting the PI3K/AKT pathway. Why? Well, again, basic science informing clinical research. A few years ago, this group published mouse model in which they proved that adding a PIK3CA mutation to BRAF-driven papillary thyroid cancers in mouse models led to progression to anaplastic thyroid cancer phenotypes and to a strong decrease in survival of this. Now, when we look at this group compared to the two others in our patient cohort of papillary thyroid cancers driven by BRAF mutation, we observed that mutations in specific effectors of the PI3K pathway, PIK3CA, AKT, PTEN, etc., well-known oncogenic mutations, were able to predict mortality. And that's what we are showing here in this progression-free survival and disease-specific mortality Kaplan-Meier curves. The red line represents the BRAF and PI3K pathway mutated tumors from patients. And in multiple regression analysis, we also observed that these mutations were independent predictors of mortality. So, recap from this part about genomics and thyroid cancer progression. Key genetic alterations can predict more aggressive clinical pathologic behavior of differentiated thyroid cancers. And I gave you two examples. One, third promoter mutations, good biomarkers of poor prognosis, especially with in combination with BRAF or RAS. And second, I just show you that mutations in the PI3K pathway can be independent predictors of disease-specific mortality in a cohort of BRAF mutant papillary thyroid cancers. And I think these results can help prospectively inform clinical cancer risk and disease outcome. Now, for the second part of this section, I will briefly mention how genomics, I think, can help inform or predict responses to radioiodine therapy and redifferentiation approaches. As you all know, radioactive iodine is being used in thyroid cancer ablation for over 80 years. And this is based on the ability of thyroid follicular cells, thanks to the expression of this sodium iodine symporter, to capture iodine from the bloodstream. And this is a feature that was used to implement radioiodine treatment, right? So this has been a story of success, I think, even although I think it's also fair to say that in the last couple of decades, the amount of radioiodine that is typically given to patients is being, more targeted and more selective and reduced overall compared to previous years. But if you think about it, it's a little bit of a paradox in the sense that we know from basic biology that NIS expression and the expression of other thyroid differentiation genes are positively impacted by the TSH receptor AC PKA signaling pathway. At the same time, we also know that the MAP kinase pathway, which is, as you know, activated in 80–90% of thyroid cancer tumors, inhibits the expression of NIS. So in a way, we are trying to use this in cells that express NIS, but also because of the activation of the MAP kinase pathway, tend to reduce the expression of NIS. Now, this recent study, so the question here is whether we can predict responses to radioactive iodine treatment. And in this recent study published in Clinical Cancer Research last year, they did exactly that. They put together a very robust cohort of patients which were classified by their structural responses to radioiodine treatment. And they classified those as exceptional responders, or ER, and non-responders, or NR. And they did an extended sequencing panel on these patients. And these are the main takeaways of the study. First, that BRAF V600E was clearly enriched in non-responders. And these mutations were completely absent in exceptional responders. Second, that non-BRAF V600E, including RAS genes and other what we call class 2 BRAF mutations, were enriched in exceptional responders. And beyond BRAF and RAS, that mutations in other genes, including effectors of the PI3K pathway and of the messenger RNA splicing pathway and the SWI/SNF complex, were also enriched in non-responders. So a good way of using genomics to stratify, predict responses, and maybe inform future use of radioactive iodine treatment. Now, going back to this pathway, an obvious conclusion of this inverse correlation between MAP kinase and TSH signaling in the effects on expression of NIS is blocking the MAP kinase pathway, right? And that's the whole idea behind redifferentiation approaches. So redifferentiation was first proven in a mouse model, in an inducible mouse model of BRAF papillary thyroid cancers, in which, as you can see here on the red arrows, I'm pointing at the expression of NIS, of the sodium iodine importer. Genetic induction of BRAF mutation suppressed the expression of NIS, and when they switched off the expression of the oncoprotein, NIS and other genes involved in thyroid metabolism went back up. Whole idea of redifferentiation, right? And that correlated with the radioiodine uptake in the mouse thyroid after switching off the gene. This could also be mimicked by using a RAF inhibitor. So not only the genetic blockade, but also a pharmacological blockade of BRAF restored radioiodine incorporation. Now, this applied to a clinical trial resulted in this first redifferentiation trial in which MEK inhibition was proven to restore uptake in a subgroup of radioactive iodine refractory patients. And even though the cohort is relatively small, you can start seeing some hints here on how genotypes can inform redifferentiation trials. All of the NRAS mutant patients responded to this treatment, but less than half of the BRAF mutant patients did so. There has been some successes with redifferentiation trial in BRAF mutant patients as well, using different drugs, and sometimes these trials are different to compare because they are using slightly different compounds and patient cohorts. But there's a little bit of everything out there, which is summarized in this table, and there are a couple of additional redifferentiation trials which were published in the last couple of years that were not included in this table. But you are, at least I begin to see a trend here on RAS mutant and BRAF class 2 patients being probably more sensitive to redifferentiation trials compared to their BRAF V600D counterpart. I think an important conclusion here is that testing for BRAF, RAS, and beyond is an important part, can likely improve patient selection for these approaches. Now, speaking of the beyond and going back to basic science, I mentioned these mutations in the SWI/SNF complex before, which were recently modeled in a thyroid cancer mouse model of the disease in combination with BRAF. And this is a summary slide of the results. And the main conclusion here is that when SWI/SNF complex is functional, this complex is a chromatin remodeling complex, so it is able to move around nucleosomes to expose certain regions of the genome to allow expression. MAP kinase inhibition is effective at restoring its expression. However, when this SWI/SNF complex is mutated in any of their components, the complex is no longer functional, and the chromatin compacting is higher than in normal conditions. So using a MAP kinase inhibitor to restore NIS expression is no longer effective in this. You remember this picture from before. Now imagine we take a few slices of these tracks, and this is what we get. And I want to use this to briefly comment on genotype-driven redifferentiation trials. So here I'm showing you two scenarios, one of a RAS mutant tumor and one of a BRAF mutant tumor going down this slope of dedifferentiation. after which, in the context of a redifferentiation trial, if we add a MAP kinase inhibitor here or here, we should theoretically be able to restore radioiodine avidity. Now, if we think that of the overall better responses in RAS-like tumors versus BRAF-like tumors, we can think of two different explanations for that, and we don't have absolute responses. One, that they are slightly more differentiated at baseline, and second, and these are not mutually exclusive, that they might have some intrinsic features that make them easier to redifferentiate. And this is something that we don't quite know yet, but I still think that we need to collect as much data on genotype-driven redifferentiation trials as possible. Now, the last thing that I want to mention here is the additional mutations. I think we now have enough preclinical data from the mouse model that I just showed to you, and clinical data as well on the paper published last year on exceptional responders to radioiodine therapy versus non-responders to, for instance, know that those tumors that are not only BRAF mutated, but also mutated in this Y-SNP complex are unlikely to be responsive to redifferentiation trials. So we should probably have the ability to genotype the tumors of those patients in advance and exclude those patients from these kind of trials and think of alternative approaches for their treatment. Recap from this part on genomics, radioiodine, and redifferentiation. It's a graphical one. So if you go RAS, BRAF, and beyond on your genotyping for patients which are candidates for radioactive iodine treatment or redifferentiation trials, I think it's reasonable to think that we can identify some patients that are likely going to benefit from these treatments and some others that should be excluded from these approaches. Now, the last 2-3 minutes, I will briefly mention an obvious use of genomics in clinical care, which is the use of targeted therapies. And I think there have been other speakers on this venue that are much more qualified to do this than myself. But just briefly, this table from our review summarizes the FDA-approved drugs for follicular cell-derived thyroid cancers. I think it's fair to say that we are beginning to move from multi-RTK inhibitors to recent approvals of highly specific inhibitors, depending on the specific alteration driving some of these tumors. Couple of examples that you are probably aware of. First, the use of RAF and MEK inhibitors in the neoadjuvant setting for the treatment of BRAF V600E anaplastic thyroid cancers, like blocking the MAP kinase pathway at 2 levels, BRAF and MEK, and showing some quite spectacular responses, again, particularly in the neoadjuvant setting. And second, the approval of I think in 2020, two RET inhibitors were approved for the treatment of thyroid cancers with RET PTC rearrangements, and how the responses also were quite good, particularly for papillary and poorly differentiated thyroid tumors with these specific alterations. So the last recap from this part, and this is also a figure that I took from our review. These are recommended and some more investigational approaches for the treatment of certain types of thyroid cancers. We'll start on the right with the red tracks representing the recommended approaches for the treatment of anaplastic thyroid cancer patients, for whom a very fast management of them by a multidisciplinary team is recommended. A quick identification of an actionable alteration, either in a tumor biopsy or a liquid biopsy, is essential, ideally by next-generation sequencing, or for a fast turnaround via PCR-based methods, or even for the case of BRAF V600E by immunohistochemistry. And then the importance of quickly matching these patients based on their mutations with the FDA-approved targeted therapies. The best results for these for ATC patients have been obtained for RAF and MEK inhibitors for BRAF deficiency mutant patients in the neoadjuvant setting. So that this would be followed by surgery and other treatment modalities. Non-responders, and I would say there is recent data suggesting that a combination in the context of a clinical trial of targeted therapy and immunotherapies are probably the way to go for anaplastic thyroid cancers, especially immune checkpoint inhibitors. Now, on the left, we have more investigational redifferentiation regimes. Ideal patients for this are slowly progressing radioactive iodine refractory or metastatic papillary, follicular, or poorly differentiated thyroid cancers. Again, trying to figure out which is the driver mutation of these tumors is the first step, and then matching with a targeted therapy, as I mentioned, for ATCs. In the case of RAS mutations being present in these tumors, an off-label MEK inhibitor could be administered, and all of these approaches could be followed in the context of a redifferentiation trial after a few weeks of treatment, if the patient becomes radioactive avid, a dose of radioactive iodine could be tested. So conclusions. I think I showed you that the genomic characterization of thyroid tumors is an essential tool for the diagnosis, prognosis, and treatment of thyroid cancers, in particular for the most aggressive forms. And I think that the translation of genomic knowledge to clinical practice requires two things, in my view. One, precise understanding of the mechanisms that those mutations activate in thyroid cancer cells. And second, clinical studies in well-defined patient cohorts. My last slide, probably to encourage discussion, are some of what I think are challenges in genomics-driven patient care. One, the resistance to targeted therapies. This is something that eventually occurs, and there are good studies out there in which genomics can help. For instance, BRAF mutant tumors under RAF inhibitors developed resistance via the acquisition of RAS mutation, a way of circumventing the MAP kinase pathway blockade, and maybe combinatorial therapies could be assessed in those cases. Second, non-actionable mutations. Most of the mutations, this is true for every cancer, not only thyroid cancer, involved in thyroid cancer progression are, as of today, not targetable. We will eventually get there. Third, dealing with tumor heterogeneity, both temporal after a sequence of treatments and spatial within the same tumor exist, even though thyroid cancers are more or less simple tumors. And we have some early data on single-cell sequencing approaches, for instance, tracking the evolution from papillary to anaplastic within the same patient and how the transcriptome of those cells changes dramatically. And finally, the inequity in access to genomic technologies or the latest developments in treatments, depending on location or in clinical care settings, which are something that is obviously important to tackle. Stop here. I saw you some data from my lab, so I just want to acknowledge past and present members of the group. Obviously, funding sources and my contact details are there, and happy to take any questions. Thank you.
Thank you, Iñigo. I just want to be really brief and say congratulations on this really very clear and enlightening review that you've given us. Before I go to any of my questions, I want to give Dr. Smith a chance to ask any questions or do any comments on your presentation.
Yes, just wanted to echo what a great publication, so eloquently written by you, Dr. Landa and Dr. Cabanillas. I really got a lot of pearls from the basic science portion of it, just how the BRAF mutation does not respond to the negative inhibitory pathway that perhaps a RAS mutation would, and how that's a stronger driver. Just more clarification about the oncocytic type of thyroid cancer, how it's more involved with mitochondria and the haploidization. I think particularly it's interesting that the BRAF V600 V600E mutation might not necessarily respond that well to these redifferentiation therapies. So currently in our 2015 ATA guidelines, when we have a intrathyroidal 4-centimeter thyroid cancer that does have a BRAF mutation, that either puts it in an intermediate or low risk category, meaning might not receive radioactive iodine, might receive radioactive iodine. Now, for me, if this patient potentially had a BRAF V600E mutation, knowing that it might potentially be more likely to be a evolve into a high grade differentiated thyroid cancer, I might be more tempted to give a radioactive iodine dose in that initial treatment, knowing that down the line it might not respond that well to a redifferentiation therapy, as opposed to, for instance, a BRAF K601 mutation that you also noted in your paper that might respond better, as well as RAS mutations to redifferentiation therapy. So yes, thank you so much. This was a great publication. It was a joy to read.
Thank you. Nice. It's tricky. I would stress that the beyond part of my presentation. So in a way, when we classify BRAF and RAS, we are missing a lot of information, right? So on the example, for the example that you just gave, in an ideal scenario, and I know this is difficult in some clinical settings, you might want to know what else is in the genome of those tumors to properly predict the next step in treatment, right? Because this is also true, and this is a little bit of a paradox, that there are also papillary microcarcinomas that are BRAF mutant predominantly, and for which active surveillance is often recommended in some clinical settings. So I would like to know on every single, this is the scientist in me, and I know it's not feasible in some cases, but for every cohort of BRAF mutant thyroid cancers, which ones are just BRAF and which ones are BRAF and something else, and plan accordingly. And I think we'll eventually go there as genomic sequencing technologies are being more widespread and cheaper by the day. So yeah.
To echo that, I wanna congratulate your team's work. Dr. Papa's presentation data that you presented on the BRAF plus, I'm gonna call that BRAF plus mutated genes. It's very interesting, and you almost had me sold for doing molecular genetics in every single patient that I see. Of course, like you say, it's a little bit more difficult than what one would want. And I want to sway the discussion in a different direction just a bit. We know, and we've heard about the use of molecular diagnosis to know which patient should go into surgery. The typical indeterminate thyroid nodule, should you operate, should you not operate? We know the use of molecular diagnosis in using targeted therapy, for example. But now let's think about something different. And I know this is going to resonate a little bit on other physicians, other specialties, especially surgeons. What about the use of molecular diagnosis on the extent of surgery? And you briefly mentioned pediatrics. I know we are focusing on adult mutated thyroid carcinomas, but the idea of that marble race, or that marble track that you presented, in a pediatric patient, the race is longer. And the time to have different hits, if I may, I know that's not a proper word to say in genetics, but the different milestones in the genomic alterations is longer, and potentially drawing one single genetic test in a patient does not exemplify the whole track that the patient's going to be going down the road. What do you think?
Well, I think that's a very good question. I think that pediatric thyroid cancers are a different ballgame, and the meaning of genomic hits in childhood cancers are slightly different than the meaning in adult cancers, for reasons that we don't quite know. And when I speak with, I'm by no means an expert on pediatric tumors, but when I speak with colleagues here at Boston Children's treating those kids, they typically do well. So sometimes you have some genomic hits that might predict for bad prognosis in adults, and in kids, they are usually fine typically after surgery. But that's something intrinsic of childhood cancers, probably biology related, that we don't quite understand yet, or at least I don't understand yet. You might have to bring someone smarter than me to that question.
There's probably a couple of people in the audience thinking that they could potentially answer those questions better than we could in this short amount of time. But what do you think? There's many questions that I want to ask, and you spoke about the TCGA project and how we find many mutations. We, however, don't know a lot of the patterns of inheritance of these genes. And in the 10,000 feet view, thyroid cancer is so indolent that if we miss the target, we still get the prize. People do well with whichever treatment we give them. But do you have any thoughts on what happens in, for example, cohorts of familial non-medullary thyroid cancer? Do we know a little bit of how these behave in family settings?
And you mean in non-syndromic familial aggregation cases?
Exactly.
Yeah, it's tricky because there is a strong familial component, but it's not Mendelian. It's multifactorial. And back in the day, I was actually involved during my thesis on gene and wide association studies. So there are multiple hits of germline variants conferring a very robust risk, but also a very small risk. So there are so many hits that we don't know because it's hard to have enough statistical power to that at this point, there is very little that we can do on that front. There are some hints of specific genes having a germline and a somatic component in certain subgroups of thyroid cancers. An example that comes to mind is DICER1 mutations, which they are more prevalent in pediatric thyroid cancers, and they were, I think, originally discovered in follicular pattern thyroid tumors, but now we've seen those in pretty much every single thyroid cancer type with a very specific pattern. The somatic mutation typically affects one region of the protein that is very important for its function. And there is usually a second hit, which is actually the first hit, which is a loss of function mutation on the germline allele, on the other allele. So that's an example of some familial aggregation or germline influence that we can easily track on those tumors.
Beautiful. Dr. Smith, I don't know if you have any further questions or comments. If not, I want to thank you both for spending an early morning here with us. Thank you for such an incredible presentation and discussion. And I want to invite you all to be here back with us next week on Diagnostic and Therapeutic Approaches to Lymph Nodes in Thyroid Carcinoma by Dr. Marco Alvarez. Hoping that will be another very interesting session. Thank you all again. Be safe and have a happy weekend.
Thank you.
Thank you.